right, this is a terrific crowd, especially for fall break. And I know we're not supposed to be on break, but still. Believe it or not. Okay, so thanks everybody for coming, and this is of course one of those really wonderful times when we get to hear from one of our colleagues. And so this will be Nadia today. And so just a little bit of background on Nadia. So Nadia got her bachelor's degree in biology from Harvard University. And from there she went on to Stanford University and worked with Dmitry Petrov and started exploring questions in molecular evolution. And I think interestingly, if you read over her CV, she also picked up an Excellence in Teaching award there at Stanford. From there, she moved on and did a postdoc at Cornell University and continuing on in evolutionary genetics and working with Andy Clark and Chip Quadro there. And then after that, she joined the faculty here in the Department of Genetics in 2010. And so she has built her program here, focusing in evolutionary genetics, and particularly in the area of influences on a really fundamental and basically important process of genetic recombination. So genetic factors and environmental factors and how those two interact. So she's received significant support from the National Science Foundation for her work. Actually just recently she published a really nice paper in the journal Science, and it's received, I think this is neat, it's received a lot of attention from the scientific community, but also science media community. I saw a nice link in National Geographic about it. And so this is part of Nadia's consideration for promotion tenure in the Department of Biological Sciences. And you're not here to hear me, so I will let her take it away, and you have your title on this. So,
Thanks very much, everyone here. Thanks for that introduction, and thank you all for coming. Really beautiful day outside. I'm sorry that you're inside, but I'll talk as quickly as I can so we can get out of here. Warming up my crowd. So if you've been outside today, or really on any day in the history of time, you probably will have noticed that variation abounds in nature. This is really obvious at the level of interspecies diversity. So there are millions, literally millions of species that live on this earth, and they're all really different, okay? They look different, they live in different places, they eat different things, they behave differently, they interact differently with different kinds of organisms. There is an incredible diversity of form and function on this earth that's obvious when we look around. One thing that's really important to understand is that incredible diversity that's so obvious at the species level is actually recapitulated at every level of biological organization. So even if we look within species, for example, we can still see pervasive natural variation in population. This is an example of a single species. This is a clam, saltwater clam. It lives in the southern Atlantic and Gulf shores from Virginia to the Caribbean. And you can see, even over this very small geographic range, we see quite a diversity of color and pattern on the shell. So even within species, with gross morphological traits, it's very obvious that species differ within and between individuals within a single species. We can zoom in even closer and look at natural variation within single individuals of a given species. So one of the most obvious examples is how organisms change form and function across developmental stages. This is a single individual that looks very differently, behaves very differently, just at different stages in its lifetime. And while developmental plasticity is a very kind of obvious example of how an organism might differ across its lifetime, there are other environmental cues that can trigger really dramatic changes in phenotype at an organismal level, again, within a single individual. So many of you are probably familiar with the example of a hydrangea. This is a single plant with incredible color variation in the different inflorescences. And the color of the petals in the hydrangea are dictated by how much aluminum that part of the plant is uptaking. And the aluminum, in turn, uptake is determined by the alkalinity of the soil. So in response to the kind of soil these different plants parts are exposed to, you can get really dramatic color variation in a single genotype, in a single plant, over a very small space. So whether we're talking about the color of a hydrangea or the size of a butterfly developmental stage or limb length or gut length or expression level or mutation rate, all of the things that we care about in this room, all of the things that make organisms what they are and function as they do. These traits are variable in nature. And one of the central goals of modern biology, and one of the central goals of evolutionary biology in particular, is to understand the origin of this variation, how this variation is maintained in natural populations, and what the consequences of that variation are for organismal fitness and function. And so what I'm going to tell you about today are our efforts over the last 5 years to understand the causes and consequences of variation in a trait that is central to organismal fitness. And that is recombination rate variation. So I'll tell you a little bit about what recombination is, and I'll hopefully convince you that it's important. So meiotic recombination simply refers to the exchange of genetic material between homologous chromosomes during meiosis. And this is an illustration from Thomas Hunt Morgan's 1916 book, illustrating how he thought genetic recombination took place during meiosis. And I show this because this figure from 1916 bears incredible resemblance to the kinds of illustrations that we use today in our textbooks to illustrate the process of genetic recombination. Now, recombination is really critical for organismal fitness, and it's critical in two regards. First, in most sexually reproducing species, at least one recombination event per chromosome is required for proper chromosome segregation. If you have recombination in the wrong place or an improper number of recombination events, you end up with gametes that have the wrong number of chromosomes. That's called aneuploidy. And aneuploidy is the leading cause of miscarriage, developmental delay, and developmental defects in humans. So aneuploidy is a really bad thing, and recombination prevents it from happening. Turns out recombination is also critical for fitness in a different way, and that is in both humans and flies, it's been shown that the frequency of recombination is correlated with reproductive output. So it's really important for organismal fitness. How many crossover events you have and where they're placed on the chromosome is central for the preservation of genomic integrity between parents and offspring. And in spite of that critical function, in spite of the devastating consequences of incorrect recombination, rates of recombination are hugely variable, right? So it's a critical process, and from an evolutionary perspective, you might think that that would mean that it would be highly constrained, finely tuned. And that's not what we see in nature. This trait, like all the traits I talked about before, they're variable. It's variable at all scales of biological organization. So we can look at species diversity in recombination rate. These are data compiled from a study from 2000, a review from 2007. This is the frequency of recombination normalized per unit base pair on the y-axis, and we have several different taxonomic groups on the x-axis. These are the social hymenopterans that kind of have outrageously high rates of recombination. No one really understands why. These are other hymenopterans, insects, and invertebrates. And across taxonomic groups, you see orders of magnitude fold variation in rates of recombination. But even within groups, we see huge amounts of variation in the frequency of crossing over as well. And again, these differences between species you can observe within species. So this is a study that was done by Chad Hunter in the lab. He looked at rates of crossing over on a particular interval on the third chromosome in 10 different strains of Drosophila melanogaster from a single population here in Raleigh. And you can see, even among these 10 strains of the same species, we see variation in the frequency of crossing over. And again, we can zoom in even further and look at within an individual. Is recombination rate variable along the chromosome? This is work we did in collaboration with Eric Stone. Here again, we have recombination rate on the y-axis. We have physical position on the X chromosome on the x-axis. And we can see that there are regions of the chromosome that have lots of recombination events, and we have regions of the chromosome that have very few recombination events. And in spite of the fact that this trait is really important, in spite of the fact that we've known that it's variable for quite some time, we actually know very little about why this variation exists, how this variation is maintained, and what this variation means for organisms. And at the center of my research program really is trying to understand the causes and consequences of recombination rate variation. And we have known for a long time that there are many different components or sources of variation Some of this is due to genetics, some of this is due to environmental conditions, and we're learning now that there's an interaction between genetics and the environment that contribute to recombination rate variation in natural populations. And work in other systems has shown quite clearly that epigenetic factors mediate recombination rate variation as well. Now, to address the individual and joint components, contributions of these factors to recombination rate variation, I primarily worked with two genetic model organisms, Drosophila and melanogaster. We do a little bit of work in mouse as well. And we use a suite of techniques, either that are specific to each system or combined across both systems, to gain insight into the role of these forces in generating recombination rate variation. The other part of our lab's work is to try to understand the consequences of recombination rate variation. Why does this variation matter? What is it doing in the genome? And we use a combination of bioinformatic approaches and comparative genomic approaches to address that. And so what I'm going to do today is tell you just a couple stories that highlight different parts of this picture. And I'm going to start by telling you about the genetic architecture of recombination rate variation. So recent work has implicated a number of genes in humans and mice that contribute to population level variation in recombination in humans and mice. Drosophila, of course, being the perfect model that it is, lacks all of those genes. And so it turns out for that and many other reasons, we actually know nothing about the genetic basis of natural variation in recombination rate in Drosophila. And so we wanted to ask, what are the genes and alleles that contribute to population and variation in this trait? Where does this variation come from a genetic perspective? Are there master regulators of recombination rate? If you have this allele, you have a lot of recombination, and if this allele, you have a little. What determines how much recombination a genome has in Drosophila? So we wanted to go into this in an unbiased fashion. We didn't want to limit ourselves to particular pathways or particular gene networks, and so we decided to use a blind approach, an unbiased approach called association mapping. And with association mapping, you're essentially correlating phenotypic data with genotypic data and asking whether particular variants of particular loci are correlated with your phenotype. And so you need two kinds of data for association mapping. You need phenotype data, and you need genotype data. And the genotype data that we were using for this association mapping project is an incredible resource that was developed in Dream Kids Lab at NC State called the Drosophila melanogaster Genetic Reference Kit. It was published in 2012. It was the first release. And what they did was, in 2002, they went to the Raleigh farmers market and they collected female Drosophila melanogaster. So female Drosophila store sperm. These females are already inseminated. They bring the females back to the lab, and each female starts, she's a foundress. They start an isofemale line. So all of the progeny are a descendant from that single female. And each of those isofemale lines is subject to full SIB inbreeding, so brother-sister mating, for 20 generations. And what that inbreeding does is it removes genetic variation within each isofemale line. So at the end of 20 generations of inbreeding, you end up with a strain of Drosophila melanogaster that's derived from a single female that is essentially homozygous across its entire genome. And that's a really powerful genetic tool because it's immortal. And there's no heterozygosity, which makes it easy to sequence. But the power of this panel is that across all the lines, and they made 205 of them, the genetic variation that's encompassed by this panel is representative of the genetic variation that was segregating in the population at the farmers market. Okay? We've minimized genetic variation within lines, which makes it a really useful tool, but we preserve variation across lines, which means we can correlate phenotypic variation with genotypic information. And that genotypic information comes from the fact that these lines are fully sequenced. And the second release, the CGRP, about over 5 million variants were called. Okay? These are variations that are segregating in nature, natural language. So those are the genotypic data we use for this project, and so now we need phenotypic data. And this project was spearheaded by Chad Hunter, the graduate student in the lab, and if you're interested in hearing more about it, you can come to his PhD defense, which is on October 28th at 1 p.m. here in this room. So we measure recombination using a classical genetic approach. It's a two-step crossing scheme. In the first cross, we take wild-type females, that's what these pluses are. They're wild-type at two different loci that we care about. And we cross these wild-type females to males. And these males are special because they're marked with two mutations, mutation 1 and mutation 2. These mutations are visible, which means we can look at the fly and we can tell the genotype. and the recessive, which means you need 2 copies of that mutation to see the phenotype. Okay? So when you cross these wild types with these double mutants, you end up with females who are doubly heterozygous for the visible markers. It's these females that recombination is taking place in, and we uncover these recombination events by back crossing these females to the double mutant males. And this cross produces 4 types of male progeny and 4 types of female progeny. And because these markers are visible and recessive, we can look at these flies and we can tell if they're recombinant or not. And recombinant individuals will have 1 visible marker, but not the other. So we can estimate the frequency of crossing over in these females as the proportion of these back cross 1 progeny that have 1 visible marker, but not the other. So that's how we estimate recombination rate. And we did this estimation for all 205 lines of the DGRP. We measured recombination on 2 different chromosomes, on the 3rd chromosome using the marker pair ebony and rough, and on the X chromosome using the marker pair yellow and vermilion. We need 3 replicate measurements per chromosome per line. And each one of those crosses involved 20 males and 20 females, and were done under highly controlled conditions. We know the environment plays a role in recombination rate variation. We want to minimize the influence of environmental variance so we can maximize our ability to detect the effects of differences in genes. Okay? So these are the results for recombination rate variation on chromosome 3R as assayed by our 2-step crossing scheme. On the y-axis, we have recombination rate. On the x-axis, we have all of the 205 strains of the DGRP. You can see the means in gray and the standard deviations are these black lines, the standard deviation across the 3 replicates. Chad scored about 220,000 flies for these line means and variance estimates. These lines are sorted by recombination rate, so the lowest recombination over here and the highest recombination over here. And you can see from these data that recombination rate is variable in the DGRP. And in fact, it varies about 4-fold from the lowest recombination line to the highest recombination line. The X chromosome looks very similar. We sort a similar number of flies. Again, these lines are sorted in ascending order of recombination rate. And there's quite a bit of variation, about 3 to 4-fold variation from the line with the lowest recombination to the line with the highest recombination. So now we have these phenotype data and we can start to ask some interesting biological questions. Okay? And the first question we wanted to ask was, are recombination rates on the X chromosome and the 3rd chromosome correlated? Okay? And the answer is absolutely not. Okay, so this is recombination rate on the 3rd chromosome. This is recombination rate on the X chromosome. Each one of these points is a strain. And you can see there's no relationship whatsoever between the rate of recombination on the X chromosome and the rate of recombination on 3R. So knowing something about this trait tells you nothing about this trait. There's no predictive value whatsoever. They're independent traits, which tells us, it's our first insight into the genetic architecture of this trait. These are 2 different traits. They're controlled by separate genetic names. This is our first insight into how this is regulated in Drosophila. Now, our goal was to find the genes and the alleles that contribute to this variation in these traits, apparently these 2 different traits. And so we used an association pipeline that was developed as part of the DGRP to identify loci and allelic variants that correlate with variation among strains in recombination on 3R and with respect to recombination of X chromosomes. And these results are summarized in Manhattan plots. These are the data from 3R. These are the data from the X chromosome. I'll walk you through this. Each one of the points on this plot is an allelic variant, a position in the genome of Drosophila melanogaster that is variable in the DGRP. They're ordered by chromosome. This is the X chromosome from one end to the other. These are the other major euchromatic arms, and this is the dot chromosome, which is a very small, mostly heterochromatic chromosome. On the x-axis is the negative log of the p-value of the correlation between the sequence variation and the phenotypic variation. And everything above the kind of nominal threshold of 10 to the minus 5 is a sequence variant where there's a significant correlation between the phenotypic variation in recombination rate on 3R here, or X here, and the sequence variant. And there are a couple things that I want you to take away from this talk. The first is that, as we saw, there's no correlation between recombination rates between X and 3R, which means that the variants that are associated with variation on 3R in recombination should not be the same as the variants that are associated with variation in recombination on the X. And that's exactly what you see, right? These kind of points don't overlap between the top and the bottom panels. And that's because they have separate genetic architectures, which is exactly what we're seeing here. The other important feature of these data is that we're seeing a lot of evidence for transregulation. So recombination rate on 3R is associated statistically with sequence variation that's not limited to 3R. There are variants on the other chromosomes that appear to modulate recombination on this chromosome. And the same thing is true for X. The variants that associate with phenotypic variation and crossing over on X chromosomes are not restricted to the X chromosome. So we ended up doing the association mapping in several different ways, and I'm happy to explain the reasons for that later if anyone is interested. But what we ended up with was a prioritized list of 20 genes that we wanted to validate further. And on this list of 20 genes was not one gene with a known function in recombination or meiosis. And that's the excitement of using association mapping, because you have this power to uncover new genes that participate in traits that are already well described. That's the excitement. The fear is that they're not real. So you can't just stop and say, I found my 20 and I'm done. We're not done. We have to show that these associations go beyond statistics. And so we decided to do 2 levels of functional validation. And one is a gene level validation, motivated by this kind of conceptual framework. If a gene participates in a given phenotype, then if you perturb that gene, you should see some perturbation in the phenotype, right? So for these top 20 candidate genes, we looked at mutant lines, lines that are missing the gene, lines for which the expression of that gene has been knocked out. And we asked, in that line, if we measure recombination using the same crossing speed, using those that 2-step crossing speed with the markers on 3 or the marker net, if we look at recombination in our mutant or in our knockdown, is recombination significantly perturbed? And that would validate at a gene level a role for that gene in our phenotype. And so we used a variety of collections of mutants. These are deletions and PLM insertions. We have the knockdown lines as well. Each one of these collections has a different genetic background control, and so, these are grouped by color. And the first one in each of these different colors is a genetic background, and then the ones that follow are the knockouts and the knockdowns. And so you do pairwise comparisons between each gene and the first one in the category. These are box plots, so the black line in the middle is the median, the edges of the box are the first and third quartiles, and then the whiskers extend to the largest point that's a non-outlier. And we've noted significant comparison with these asterisks. And you can see when we look at recombination on 3R in these mutants and knockdown lines, there's only one gene that shows a significant change in recombination frequency when you perturb the gene at a molecular level. Now we do a little bit better when we look at recombination on the X chromosome. Quite a number of deletion lines and PLN insertions show significant decreases in recombination relative to the control or increases in recombination relative. So overall, about half of our 20 genes, either on 3R or the X, show some perturbation in recombination frequency when you perturb the gene. But that's only one level, right? Does this gene function in this pathway? And for some of our genes, we do see gene level validation. But we're really interested in understanding whether variation in that gene, whether allelic variation in that gene is associated with phenotypic variation in our trait. And so we're not done when we get to gene level. We have to look at allelic differences. Does this allele produce something different than this allele? And we hypothesized that if there were allelic differences, they'd be mediated by expression level differences. And so to test for allelic effects for all 20 of these candidate genes, we looked at expression level of each gene in the ovaries, comparing the expression of the major allele to the minor allele. And so Chad basically selected 3 GGRP strains that had minor allele and 3 strains that had the major allele for each of our 20 candidate genes. We have the major allele here in blue, and the minor allele in red, and this is the expression level specifically in the ovaries. And you can see again about 2/3 of the genes show significant differences in expression between the major and the minor allele. And so if you integrate these 2 levels of validation, the allelic level and the gene level, what you end up with is 4 high-quality candidates that we'd like to pursue further molecularly. And these are the genes CG10864 and CG33970. We don't know very much about these genes, but we're computationally predicted genes. This gene here is an excision-induced protein, 75B. We're excited about this one in particular because it has a known function in oogenesis. And then the last one, P2P61F, is a protein tyrosine phosphatase. And so we're, in the future, going to look at the molecular level at all these genes to see, to further validate their role in meiotic recombination and meiotic recombination variation. What I've told you so far is that our study of recombination in the DGRP has revealed 3 to 5-fold natural variation in frequency of crossover on 3R and X chromosomes. What I didn't tell you is that of that phenotypic variation, about 15 to 30% of that variation can be ascribed to genotypic differences among strains. We found, I think very interestingly, that the recombination rates on chromosome X is decoupled from recombination on chromosome 3. Our association mapping revealed a number of candidate genes and alleles that associate significantly with phenotypic variation in recombination rate. And we were very excited that all of our top candidates were genes that had no previously documented function in homologous recombination or meiosis. And we did validation at 2 different levels and integrating across those avenues of investigation yield for high-quality candidates for future functional assays. And so I've told you a little bit about our efforts to understand the genetic architecture of recombination rate. And I'm going to switch gears now and tell you about some recent work about the effects of environment on recombination rate. And I want to kind of explain to you where we, our perspective, we're coming to this problem. So recombination is a funny thing. Recombination creates new combination of alleles, which can be a very good thing if you have a bunch of deleterious alleles or bad alleles hanging out. So that's a good thing of recombination. Recombination is kind of not a great thing when your genotype is a collection of really good things. Recombination breaks them up, right? So recombination is constantly undoing what natural selection is putting together. And this kind of cost of recombination is an evolutionary puzzle. The fact that recombination is so pervasive in nature and is maintained in natural populations is really one of the biggest open questions in evolutionary biology. And one of the leading hypotheses for the evolution of recombination in stacks and for the maintenance of stacks in populations is that sometimes there's a benefit for offspring to look different from their parents. And that benefit is especially realized when individuals, when species, are co-evolving with other organisms like pathogens or parasites, okay? And so if you're infected with something maybe, or there are lots of things that are capable of infection in your environment, then maybe you want your offspring to look different from you so they have a better chance of being resistant to infection or tolerant of infection or something else. So it's like a genetic bet hedging strategy. And so this is what people have called the Red Queen hypothesis. It's called the Red Queen hypothesis because the name is taken from the Red Queen character in Lewis Carroll's Through the Looking Glass. And the Red Queen says to Alice, it takes all the running you can do just to stay in the same place. The Red Queen hypothesis for the evolution of recombination describes this co-evolutionary dynamic between hosts and pathogens such that there's an evolutionary benefit to diversifying your offspring such that they have a better chance of evading the pathogens and parasites that are in the environment. And this hypothesis has been around for quite some time. And we wanted to take it a little bit further. So we're very interested in recombination plasticity, so the capacity of a single genotype to produce a different phenotype depending on the environment. And there's this huge body of literature about the evolution of plastic recombination. But we wondered whether the evolution of plastic recombination could be due to the same pressures as the evolution of recombination in the first place. And so we wondered if we could extend this Red Queen idea to plastic recombination instead. So the idea is this: if recombination is beneficial over asexuality in populations when there are parasites, then maybe within an individual who has recombination machinery already, maybe when an individual is transiently infected with a pathogen or a parasite, maybe recombination rates will transiently go up also. So it's a very simple hypothesis, but no one has really put together plastic recombination and recombination in the same conceptual framework, and that's what we did. And so we wanted to ask, if we transiently infect Drosophila with something, will rates of recombination go up? So we measure recombination using the same scheme that I told you before, the two-step crossing scheme. And we're focusing on recombination that happens in these doubly heterozygous females, and now we're just going to infect them, right? We're going to infect them with something and see, does the proportion of progeny that is produced by these infected mothers have a higher proportion of recombinant progeny, okay? And so the first experiment we did was to just infect adult females with a bacteria called Serratia marcescens. This is Serratia marcescens. It has this really beautiful color polyphenism. Depending on the temperature you grow it at, it's either white or red or something in between. Serratia marcescens is a gram-negative bacterium. It's an opportunistic pathogen of humans. It has an incredibly wide host range, infects Drosophila in nature. And we infect these doubly heterozygous females by taking a very small tungsten pin. We dip it in a liquid culture of Serratia, and then we just poke the flies for it. And the control here is that instead of dipping it into liquid culture, we dip it into media. So we just ask, if you infect flies with Serratia marcescens, does the proportion of progeny produced that is recombinant increase? And the answer is yes. We used 4 different strains for this experiment. The small Drosophila melanogaster, again, they're taken from the VGRP. This is the recombination fraction. This is the proportion of those progeny that is recombinant. We have our sterile wounds in the dark gray, and we have the infected with bacteria in the light gray. You can see that the proportion of progeny that's recombinant increases significantly, plastically, in response to bacterial infection. So we were really excited by this finding. No one had seen it before in an animal, and we wanted to get a little bit deeper in terms of how this works, in terms of mechanism. And we hypothesized that there were kind of 2 pathways by which this could occur. One is, maybe when flies are infected, something happens with how crossover sites are designated or how they're resolved. Maybe you get more breaks. Maybe more of those breaks are resolved as crossover events. And that's how you get more recombinant progeny. You still have to have more recombination events happening in meiosis. The other possibility is that there's some sort of non-Mendelian transmission of recombinant versus non-recombinant progeny. We're going to call transmission distortion, that something else is happening such that recombinant individuals have a fitness advantage in some way. And we wanted to distinguish between these 2 hypotheses, and we're very fortunate to say Drosophila because their biology actually makes distinguishing between these 2 alternatives possible. And that's because of the way that oogenesis works in Drosophila. So Drosophila has ovaries, a pair of ovaries, and each one of the ovaries is made up by a number of ovarioles. And these ovarioles are essentially assembly lines for the production of oocytes. Okay? And so the production kind of starts down here in the germarium, which is kind of blown up here. And then the process continues along the ovariole until eggs are fertilized and mated. Now, crossing over, recombination, it happens very early in this process, early in the germarium. And in fact, it takes 4 or 5 days between when recombination happens here and when that egg can be fertilized in the lake. So if infection is doing something to affect the process of crossing over, how many breaks are made, how those breaks are resolved, then if we infect flies, we shouldn't see any effect for 4 or 5 days, because that's how long it takes to get from here to here. But if we see something happening sooner than 4 or 5 days, this strongly indicates transmission distortion. So we exploit this biology to gain insight into mechanism. And we did a slightly larger experiment. Now we have 4 different treatments. We have a no treatment control. We don't do anything to the flies at all. We have our sterile wound, our old friend Serratia marcescens, and then we try the second bacteria just to see if other types of bacteria can elicit a similar response. This is Providencia bruckeri. It is another gram-negative bacterium. It's a naturally occurring pathogen of Drosophila. We treat mated F1 females with one of these 4 treatments. We place individual females in vials, and then we do the serial transfer, and that's to separate those eggs that are already post-crossing over at the time of infection from those that are not. So if we see something in the first 4 days, this is our signal that transmission is being distorted. Something non-Mendelian is going on, and that's exactly what we see. Okay? This is our recombination fraction on the y-axis. We have our 4 different treatments on the x-axis. The no treatment control is unfortunately not informative because it's not significantly different from either our wound control or the bacterial control. But if you compare the sterile wound to the Serratia marcescens infected flies and the Providencia rectori infected flies, you can see the significant increase in recombination infection associated with bacterial infection that manifests early. This is too soon for these events to be meiotic in origin. This is transmission distortion. This effect persists in the later egg collection stages, and now we see significant difference between both of our bacterial infections and actually both of our controls. So we were excited by this, and then we started thinking, Red Queen isn't about bacteria, right? It's about pathogens and parasites. So if we infect with a different kind of thing at a different developmental stage, will we see the same thing? And so we teamed up with Todd Schlenke, who is at Reed College, and Todd studies wasps at parasitized Drosophila. And this is one of those wasps. This is in the genus Leptopolina. And these female wasps, this is a female wasp here, and this is a Drosophila larva here. The female wasp will lay a single egg inside a Drosophila larva. And the larva will mount an immune response. And if that immune response is successful, the larva will encapsulate that egg in a melanotic capsule. Okay? If the immune response is not successful, the wasp larva will devour the fly larva from the inside out and then actually eclose from the Drosophila pupa. But the nice thing about the system is that you can actually look at these larvae and tell who has successfully fought off a wasp attack. So we asked a very simple question. If we let these wasps attack our larvae now, do we see an increase in recombination fraction? And again, the answer is yes. These are our wasp-attacked flies, these are control flies, and we see a significant increase in recombination fraction in the wasp-infected flies. I think the most amazing thing about this result is that at the developmental stage where these flies are being attacked, they don't have ovaries. They don't have ovarials. They're not producing eggs. So whatever the wasp is doing, whatever the immune response is doing, whatever the signal is, it is sustained across development and ultimately realized 10 days later when those flies start producing eggs. So when the red queen hypothesis was first kind of formulated, it was all in the context of coevolution between host and parasites. Well, I think at its core, it's about dynamic environments. It's about changing selection pressures. And it's easy to see how selection pressures are changing when you've got a co-evolving organism. But selection pressures can change even without other organisms. And we were interested to know whether other types of dynamic environments could lead to similar changes in recombination fraction. So this work was done mostly by an undergrad in the lab, Dallas Crisco, and a postdoc, Catherine Cole, and research support specialist, Stephanie Ruja. And then this got another undergrad in the lab, Savannah Jackson, interested in other types of stressors. And so these are her data. This is a paper that we published in collaboration with Sally Nielsen, asking about temperature shock. We'd known that heat shock increases recombination, but what Savannah wanted to know was, does the duration of the exposure, the intensity of that stress, does that correlate with the magnitude of the change in recombination? So she took flies and she heat shocked them for a different amount of time, and she showed quite clearly that the longer you stress these flies, the more they respond in terms of increasing the recombination fraction. And again, these are eggs laid early in the first 5 days following heat shock, so this confirms the role of transmission distortion for mediating an increase in recombination following stress. Chad Hunter and Matt Robinson, another student in the lab, were interested in the effect of maternal age. Aging turns out to be very stressful. And it's stressful not only in many ways, but one of those ways is recombination. And so these are just 4 different strains of Drosophila melanogaster, measuring recombination as a function of that female's lifetime, and you can see that over time recombination rates increase. So what I've shown you here is our work on blasted recombination. We're very interested in stress-associated blasted recombination, and stress increases the recombination fraction in Drosophila melanogaster. Very clear in response to parasitism at different developmental stages. It's true in response to heat shock as well as maternal age. And what we're doing right now is trying to see if it's characteristic of the entire genome. So we've only surveyed one locus here, and it could be somehow unique to that locus. And so we'd like to look at the entire genome to see if this pattern is recapitulated everywhere. We're interested to see the scope of stressors that elicit a recombinational response with the hopes of gaining insight into the mechanism. We're really interested in the mechanism. How is transmission being distorted? We have a hypothesis that it's an asymmetry in meiosis II, but we really need to get to the lab and test it. One other question that we'd like to answer is, so it's clear that transmission distortion plays a role. Okay? It's not clear that meiosis crossing over in prophase is not perturbed also. So just because transmission is happening doesn't mean that something else isn't happening too. And so we want to know whether stress increases meiotic recombination during prophase. And we're eager to test this in Drosophila, but it turns out mice are a lot easier to work with to answer this question. And so we looked at mice first. This is work done by Beth Dumont in the lab and in collaboration with Jennifer Miller. And the difference with mouse that makes it really useful is that we can look at meiotically active cells. They're actually undergoing crossing over. And the crossover sites like literally light up. We can see them under a microscope. And we can count them. And so we can ask, in response to infection, does the number of crossover events in meiotically active cells change? And Beth did this experiment in 2 different strains of mouse, BLAC6 and PWD. We have 3 different treatment groups. On the y-axis is MLH1 count. That's our indicator of how many crossovers are happening in each cell. We have our infection. We used a bacterium in the genus Borrelia for our infection. We have immediate control, that's this MC, and then NO is a Nair only. So it turns out to inject mice with bacteria, you remove hair, and the most effective way of removing hair is to use Nair. So this is the Nair only control. I think they weren't playing that when they marketed that product, but that's okay, it's really useful. And you can see that there's no difference at all in the number of crossovers in these infected mice versus either control. Okay? So at least in mouse, it looks like the number of crossover events is not plastically altered in response to this bacteria. But again, we'd like to follow this question for some. We'd also like to expand this to other species and see if this kind of plastic increase in recombination fraction in response to stress is evolutionarily conserved across time. So in the last few minutes, I'm just going to tell you briefly about one little story that tries to show you the importance of recombination rate variation for the evolution of the gene. And working in Drosophila can be very useful because we can take advantage of the genomic resources that exist for this species. So that we have many minutes. There are many Drosophila genomes that have been sequenced over the last several years. This is just the group that was sequenced in the 12 Drosophila Genomes Project, which was published in 2007. And we can use these whole genome sequences to ask really simple questions like, what are the consequences of recombination rate variation? And at its extreme, we can just ask, what happens if you have no recombination? What are the consequences for the evolution of sequences when recombination is completely gone? And we have chromosomes, right? The Y chromosome doesn't recombine at all. And so we can compare patterns of evolution between non-recombining portions of genes and recombining portions of genes to get our first insight into the importance of recombination for the evolution of sequences. And so we use the 12 Drosophila's genome sequences to ask this very simple question. How does the lack of recombination affect sequence evolution and other things like the efficacy of selection? Okay, so it turns out natural selection, the efficacy of selection is dependent on recombination, and weak selection is particularly sensitive to how much recombination there is. And one thing that people have done over the last probably 10 or 15 years is they've used this thing called frequency of optimal codon. It's a metric of codon bias, and it's not important or interesting what it is. What's important is that it's a very useful indicator for the efficacy of natural selection. And when FOP is high, that's an indicator that the efficacy of weak selection is high. And when FOP is low, that's telling you that the efficacy of natural selection on weakly selected sites is low. Okay? So we're using this as a metric to tell us something about the efficacy of selection on weakly selected sites. And these are 9 different species from the 12 Drosophila Genomes Project, and each one of these points on these plots is a gene, and and the median is shown in these red bars. All the genes are on these different chromosomes, and Drosophila have these Muller elements that are labeled A, B, C, D, E. F is that fourth chromosome, which is a dot chromosome, and here we have the Y chromosome. And what you can see, if you can see nothing at all, I mean, the one thing that jumps out at you, right, is that on the Y, PHOS is consistently low. It's lower than any other chromosome except the F chromosome, which is also heterochromatic and doesn't recombine. And so one consequence of having no recombination, like these genes on my chromosome, is that FOP is really low. And again, FOP is our indicator of the efficacy of selection on weakly selected sites, which means that when you don't have recombination, selection can't do its job. And that's really glaringly obvious from this plot, but we can be a little bit more sophisticated about the comparison and control for gene function. So the Drosophila Y chromosome is really fun because for some reason that no one understands, genes kind of hop on and off of it all the time. And so across those 12 species, we have species in which a particular gene is on the Y chromosome in some species and on an autosome in other species. And so this allows us to control for gene function, right? And so we can say for this gene, which has this function, does it matter if you're Y-linked and have no recombination versus being autosomal and having recombination? And again, here's our indicator of the efficacy of each selection for our 5 genes that move all over the place. And you can see in every pairwise comparison, there's a significant decrease in FOP, significant decrease in the efficacy of selection on weakly selected sites, simply as a consequence of being on the Y and having no recombination. Okay? So these data really show that the lack of recombination compromises the efficacy of selection at weakly selected sites. And what we're doing now is asking whether this is recapitulated at a fine scale. So not yes recombination, no recombination. What about a little recombination versus a lot, or a little versus a moderate? Do we see these differences in patterns and rates of evolution at a finer scale? We're very interested in characterizing the distribution of recombination events along the chromosome at a fine scale, understanding how that distribution varies within and between species. How does it evolve? And we're trying to understand the consequences of that variation for evolution under different evolutionary scenarios, like background selection or hitchhiking. So obviously, I have not solved the problem of why we have many species on this earth in today's talk, but I hope I've been able to convince you that variation is everywhere in nature, including with traits that are incredibly important for organismal fitness, and that you can use really basic approaches to try to understand where that variation comes from and why it's important. Before I stop, I'd just like to acknowledge everyone who did the work that I was presenting today, so people who are in my lab either directly or physically. The people in blue are people who contributed to the work that I talked about today, but other members contributed in other ways. There are a lot of collaborators we had on this work that I presented from a variety of different institutions. And then a huge number of colleagues made it possible either by providing reagents or strains or space or equipment or advice or reality checks. None of the work that I talked about would have been possible without these people. I'd like to acknowledge my funding sources and Doug and Nielsen for the spectacular photos that were used in all of the press releases. And I'll just close by saying that this is obviously a promotion talk, and as such, I really want to acknowledge all of the people who have been here with me for the last 9 years, friends and colleagues, and I'd like to, there are too many to enumerate, but I hope you know who you are. And I'd really like to call special attention to the people who have paid particular attention to mentoring me and guiding me over the last 5 years. The mentoring committees, I had 2, I made a lot. Stephanie, Trudy, Eric, David, and Jeff, and then all the leadership we've had in this and other units over the last 5 years. I'm really grateful to everyone for their support and encouragement. And with that, I'd be happy to take any questions if anybody has any. Thank you for your attention.
It's a great talk. I have like 20 questions, but I'll limit myself to 2. First, if I understand your DGRP setup correctly, you're measuring the difference in recombination in the F1, so you're mainly detecting dominant effects.
That's exactly right.
So if you were to go to the F2, you might uncover an entirely different portfolio of recessive effects?
Absolutely.
Okay.
That would require everyone in this room. And if you can get them all to sign up, I'll totally do that experiment.
I'll push flies for you. And then my second question is more of a sort of a thought question. That is, with your hypothesis about how parasitism or pathogenicity affects plasticity of recombination, do you know if you look broadly at, say, asexual species who had sexual ancestors or cryptically asexual species like Deloid rotifers, do they have a comparatively lower parasite or pathogenicity load than sexual species?
That is a really great question. And I don't know the answer to that. Does anybody know the answer to that? It's a great question. I don't know if anyone has, I mean, surely someone has looked at that. Although I don't know. We do know that in species that are facultatively sexual, rates of sex go up with increasing parasite burden.
Okay.
That's clearly true. But in terms of an evolutionary perspective about how sex evolves from asex and load, I'm not aware of any work on that topic. Great question.
Okay, so I want to go back to the first part of the talk, because it's very striking in your Manhattan plot that you had all these trans factors potentially affecting combination. Because I think if the literature from the 80s, Brooks and Marx, was like more cis factors were what they were arguing was relevant. When you did the validation step, did you see a shift towards more cis factors, or did it tend to retain that heavy representation of trans factors?
You know, with only 20, it's hard to draw firm conclusions. I would say we see evidence of both cis and trans in our validation, and I would say that recent work from David Pressfield's group implicates both cis and trans in median mole combination rate, at least not in the lung cancer, but cultural. So the Brooks work from the 80s, again, it was a very limited number of strains, and it's hard to know if that was generally representative of populations.
Just one additional follow-up is something going back and thinking about stress, right? Your mouse experiment did not recapitulate what you saw in software, but you sort of make this argument that it's actually stress-driven. How can you measure that you're applying equivalent levels of stress between these? Would that be relevant?
It's absolutely relevant, and we are absolutely not applying equivalent. So there are two main differences. Well, there are a lot of differences in the mouse study and the fly study, not just mouse versus fly. One really important thing is that with the mouse experiment, we don't have the ability to look at transmission distortion. We're only looking at active meiotic components, okay? And in Drosophila, we're only looking at transmission distortion, maybe. We can't separate, but we clearly see transmission distortion. If it's all transmission distortion, we would not be able to detect it in our mouse experiment. The other thing about the mouse experiment that's really important to mention is that that bacteria that we use doesn't make the mice sick. And so if it's stress, then we wouldn't see it in an experiment either. And because there's no active immune response. And so what we would like to do in the future with mouse is actually just really stress them and see if so the lack of detectable effect in the mouse experiment does not indicate that there is no such phenomenon in mouse. It just means that this particular bacteria doesn't elicit increased presence.
Very nice. I think I would have given this talk better myself.
Wow, that is quite a statement.
I have a question about the parasites, the infectious bacteria. The offspring from the flies that were injected with bacteria and that have great recombination, are they in fact more resistant to subsequent infection? Or, if we don't know that, is it possible to take those flies and keep injecting them again over multiple generations through selection and see how far you can push the recombination rate up? And if by doing that you will actually make the spread in the recombination rate smaller over time and concomitant with increased resistance to pathogen infection. Are you doing that, or have you thought of that, or do you have any information? Because I find that very exciting.
It's very exciting. And that's really a critical piece of information to put this firmly in the red queen framework, because if it's really red queen dynamics, then the offspring of infected mothers should be more fit, more resistant to parasites. We haven't done that experiment in part because it's like two genotypes that we're combining in this experiment. So there's not a lot of genetic diversity in those progeny. It's just combinations of, in different ways, of two different genotypes. We are trying to collaborate with a group in India that has been co-evolving Drosophila with a parasite for many generations, and they in fact third generation, and we're very interested to know whether recombination has evolved to increase in the co-evolved lines. It's been shown quite clearly in Tribolium that when you co-evolve Tribolium with a parasite, rates of recombination increase as well. So people have seen it from an evolutionary perspective in terms of co-evolution. This was looking at plasticity, so how rates of recombination within a single individual change. But you certainly get co-evolution, recombination increasing in the presence of presence.
But it also comes with the question that if I look at the actual percentages different,
Yeah.
they're fairly small.
They're
So
very
the question is, how high can you, is the normal recombination rate already near the ceiling of how far you can get?
No, I think that's a really good question, and we don't.
It's very nice talk. So it's interesting that the interval on the X chromosome and the interval on chromosome 3, they're not correlated in that association population. Not correlation in the sense between the different lines, but also not correlated in terms of your candidate genes, your SNPs are all different.
Right.
So, I mean, so you're saying the genetic architecture controlling the combination of chromosome 3 in this particular interval on chromosome 3 is completely different to the genetic architecture controlling this interval on chromosome, not on the X chromosome. Presumably there are infinite number of intervals in the genome. Are the genetic architectures controlling each interval different? I mean, and in that sense, do you really care about a candidate gene that controls recombination of just one specific interval, of no particular importance in the genome? I mean, would you be more interested in things that control genome-wide recombination?
Absolutely. And if we'd known the architecture going in, we might have taken a different trajectory. But I hypothesized that there would be master regulators like we see in mammals. I did not expect that this trait would be controlled by hundreds of loci of minuscule effect. And if we did, maybe that's why no one's done this before. They had better hypothesis generating abilities than me. No, I mean, does it matter? I mean, it matters in the sense that we care about understanding how this process is regulated. So from a basic biology standpoint, building up our understanding of the process and the players in the process is important. For practical applications, like if someone going to come to me and say, man, I really wish I could change recombination between ebony and rough on 3R, I hear you're ready to talk to me. That's like not going to happen. But from basic biology, I think it's important, and the goal really, the goal was to determine the genetic architecture in the hope it was interesting, but now we have a better understanding of how it's regulated, and I think we can stop there.
But the fact that you didn't find a master genetic regulator in your process just means that there wasn't standing variation at those loci. It doesn't mean that they don't exist.
And that there are no dominant antigenic variants. Absolutely. That's in this population. That's exactly it.
I had a question. If there are anybody had looked at sort of recombination rates in insects with highly symbiotic bacteria? Do you see lower rates of recombination with these?
So one of the things that Chad found in a mapping study, so Drosophila melanogaster, many arthropod species are naturally infected with Wolbachia. And one of the things that came out of Chad's study was that we found a significant association between Wolbachia infection status and recombination rate, but only on the X chromosome. It does not affect recombination on the third chromosome, but there's a significant effect of Wolbachia infection on the X. So we have this data. We're not sure how to interpret those data. And I don't know, I think there's probably work in bees especially looking at that, but I'm not really familiar with those. Here.
So you look at a number of stresses, combination seems to increase, almost chromosome 3 as well, a number of stresses. Is it always transmission disorders, or is it,
We don't know yet, and we want to, that's the subject of like right now. We don't know. We always see transmission distortion, but that doesn't preclude other meiotic events from participating in the test, to be able to test that.
Okay, if there are no other questions, let's thank Nadia again for Nadia.